Hi everyone, my name is Peggy Hill and I'm going to be explaining the neuromuscular junction. As the word already says it, you have your neuro, so your nerve. That's your axon. And then you have your muscle, that's the skeletal muscle down here. And then the junction is what I call this in between, the synaptic cleft. So this is not attached, they're not together. Okay, so what is the neuromuscular junction? It is a chemical synapse formed by the contact between the neuron and the muscle fiber. So when the motor neuron transmits a signal, the muscle fiber, to the muscle fiber, it will cause a contraction. So that's where you get your muscle contraction. So let's start with the structure. The easiest way to remember the way it looks like is an arm and the fingers. That's how they described it in the book, and I like that pretty much. So here's your arm, forehand, and here you have your fingers. So the junctional folds would be your fingers. That's where the synapse is down there. Okay, so let's start on the top. You have your motor nerve fiber. So that is insulated with a myelin sheath as a protection. Then you have the Schwann cells right over here. It envelopes the entire junction. So this is closed and isolates it from the tissue fluid. So just think that this is the tissue fluid, the brown part. It will not enter. Okay, this part right here looks like a doorknob. This is the synaptic knob. Easy to remember. I also heard in one of the videos that it's called, or also called stamp. So whatever is easier to remember, I would say knob because it's that way in the book. And I would use it that way, not so we can confuse anybody else. But if we hear it, it's also called like a stamp. So the synaptic knob contains organelles. These organelles are called spheroidal, easier word, synaptic vesicle. So in these synaptic vesicles, they contain ACH, acetylcholine. So those are your neurotransmitters. They send out your message. So the nerve sends out your message is in here, and the message needs to go to your muscle. Okay, this will be explained in a minute. Then you have your muscle cell sarcolemma. This is the part right here. And the nice cute golden stuff, that's all around. It's called basal lamina, and it is in the cleft, so there's more gold in here. I ran out of it. So, but we can see it. So this contains ACHE. So what is ACHE? It's acetylcholinesterase. It's an enzyme that breaks down the ACH after stimulating the muscle cell. It turns the muscle contraction off, allowing the muscle to relax. Okay, so here are your receptors, the ACH receptors. They have protein in there. And I read that we have 50 million of them in our muscle. That's a lot, but I mean, we also have a lot of muscles. Okay. The receptors, before I forget it right here, those are the ion channels. They are embedded in the cell membrane that close and open when the ACH binds to them. So here are our ACH. They bind to the receptors. That's when they open their channel here and get into your muscle cell. Okay, so let's explain how this happens. So you have your signal from your brain coming down to tell you to kick the ball. Sorry, I made it a little bit too big. Okay, this opens up your calcium. Oops, I dropped one. 1 second, please. Put them down here. So I only made one calcium. So you have the calcium and the natrium. So when this begins, so when the signal goes to the axon, goes down into the synaptic knob, it starts to depolarize. That means it's reversing the charge of the cell. So I made here the calcium, I open it up, so you have like plus minus would be plus minus, so it reverses it. So this turns into the ACH molecule right here. that undergo exocytosis. This is a form of active transport. So cells transport the molecules out of the cell by the energy-dependent process. So they all get pushed down here. There you go. Okay, due to the energy in there. Okay, and now active transport, they're going to be pushed out. So X, easy to remember, X, the pseudo is the cell. The other way around would be endo, so it would be going inside. You can also think of a vacuum cleaner, but in this part, we're talking about the exo. So our ACH gets out here and starts attaching to our receptors. There we go. Da-da, da-da, da-da. And while this happens, okay, so the ACH, our acetylcholine, the neurotransmitters are now attached to the receptors. So they received it. Okay. So this allows the sodium to enter into our muscle cell. Calcium will enter too. In this case, we have a calcium induced and a calcium release, which causes the synergy. That means the muscles will contract. And when one muscle contracts, the other muscles will contract. In here in the cell, you can see it better in the book though than how I colored it. You have the sarcoplasmic reticulum. That's where you also have the calcium storage. So when you have the calcium coming into the body, it releases it from here. And that's what causes the contraction and the relaxation due to the calcium. And then that's when your ACHE starts working, your acetylcholinesterase, the enzyme that starts breaking down the ACH. It's being eaten up. There you go, all gone. And turns off the muscle contraction. So that means the muscle will relax afterwards. Okay. I forgot the nucleus down here. So the nucleus, it's dedicated, it's beneath the muscle folds. What are they called? Junctional folds, sorry. So the nucleus is under the junctional folds, and they are dedicated to the ACH synthesis and the other proteins of the sarcolemma. Okay, and I think I have, I'm not sure if I said it, but where do we have it? The basal lamina and the sarcolemma in the cleft that contain the ACH, they also have collagen and glucoproteins. So we have that too. I think I have all. Okay. Please, yes, give me input. I picked something that was hard for me to verbally express. And if I missed something or if you guys have a better way of explaining it, please do so. Thank you very much. Bye-bye.